And now, dear students, we start the third video on transport in humans, which is O-level biology 5090. And this is just a few components left of the chapter which we will finish in this video. This is the syllabus point H. This is syllabus point H. It says in the syllabus, identify red and white blood cell as seen under the light microscope on prepared slides and in diagrams and photomicrographs. So here are the ones where you can see a whole lot of everywhere. These are the red blood cells. These are the red blood cells. These are the red blood cells. So you can see the red blood cells which are in abundance. They're much more. They're many in number as compared to the white blood cells. And then of course you have the phagocytes. So these are the ones which you can see the phagocytes. This is the phagocyte. This is the phagocyte. And then you have this one which is could be labeled as the lymphocyte. So please be able to identify these. You can see, you can put in the Google search labeled diagrams or labeled photomicrographs of blood, and you can get a whole lot of picture of them, and you can practice doing these whenever you're revising this chapter. So you should be able to pick them up very easily. You can see the phagocyte has a large, it doesn't have a large nucleus, it has a lobe nucleus, like an earlobe. It's got these dots in the cytoplasm. That's why it's also called a granulocyte. While as opposed to this, the lymphocyte has a large nucleus which nearly fills the whole cell, and it's just got a slight bit of cytoplasm on the side. So please understand how you would be able to identify them on photomicrographs. Come to the prevention of coronary heart disease, which was left out in the second video. So preventive measures so naturally we're going to prevent the number one fact, which was diet. Diet was intake of saturated fats. So less intake of saturated fats, and less intake of saturated fats means less animal fats. Less animal fats means less butter, less cream, less ghee. So desi ghee, less intake of saturated fats. So in your diet, you should be having more of the plant oils, which is olive oil or corn oil. So those are good for you. Then the second factor was stress, which resulted in. So if you can reduce the stress in your life, maybe you need to change your job, maybe you need to change the environment where you live, or something else is bothering you, you need to go and fix that, or you need to just get used to it and change your lifestyle. So reduce stress. Reduce stress means reduce the causes of the stress. Whatever the cause of the stress is, you need to remove that from your life. And the third and the most important one is, of course, you give up on smoking. If you smoke, well, if you don't smoke, well, you're lucky. But if you smoke, then you better give it up on smoking. So, because smoking predisposes to coronary heart disease. Come to the F part of the syllabus, which is investigate and state the effect of physical activity on pulse rate. So basically, if I say investigate, you've got to give me a practical procedure. So, where will you check the pulse? The pulse is very often checked at the wrist, on the side of the thumb, and you can just press a little bit and you can feel the pulse very easily. So you need a stopwatch to look at, or you need a clock in the room to look at the seconds watch, and you can count the pulse in 15 seconds and then get how much is the pulse rate in a minute, or you can count the pulse in 30 seconds and then calculate what is the pulse rate, how much is the 70 to 80 per minute, right? So you first take the pulse rate at rest. So you measure the pulse at rest, and that is usually this much. And then, of course, you ask them to perform an exercise, whether you do the step test, which is you just have a small stool and you step up on it and step down. You don't need a very sophisticated apparatus. It's just called the step test. Of course, you can use the treadmill, or you can think of some other standardized exercise. You can't say just run around the room. I don't think that's not a very clear instructions that you give. So you perform some exercise which is going to be for about 2 or 3 minutes, or maybe even up to 5 minutes, and then you measure the pulse rate again. Now, usually what will happen is that the pulse rate will increase. Naturally, it will increase. So what is happening when the pulse rate increases? The basic understanding is that your circulation becomes faster. So if the heart is working, say 70 per minute, and now it's working 90 per minute, so the circulation is faster. So the oxygen which has been combines with hemoglobin in the lungs, so it's going to reach the leg muscles in faster. Maybe it used to take 4 seconds, now it's going to take 2 seconds. So oxygen reaching the muscle faster, and of course, carbon dioxide, which is being produced by the process of respiration as the person is exercising. So the leg muscles are exercising, and they are producing more carbon dioxide. So the carbon dioxide is reaching the lungs faster, and you're going to just exhale it out. So it says investigate and state the effect. So the effect of all this is that the circulation, the heart rate increasing and the circulation becoming faster. The next topic is phagocytosis. Now, phagocytosis is when the phagocyte, when, for instance, somebody cuts their skin and there's a cut, so the cells of the skin are damaged. And what we have everywhere in the body are capillaries. Now, the capillary network is everywhere in your body. So, what happens inside the capillary? You have the blood flowing, and suddenly now, when you have damaged cells of the body, they release a chemical which is called histamine, and that is detected by the phagocyte. The phagocyte moves out of the capillary, and this movement out of it is called an amoeboid movement. So the phagocyte, which was flowing inside the blood very comfortably, now decides to move out, and that is not possible for any other blood cell. So the phagocytes move out, and then they engulf the pathogen. So the is going to phagocyte is going to engulf the pathogen, and the pathogen could be what? Could be a bacteria, could be a virus, could be anything. And then it forms a phagocytic vacuole. The phagocytic vacuole means that it is going to form, the vacuole becomes part of it. Now I'll just draw it again and then you can see what I mean. And it becomes involved, it just engulfed into it. And now what it contains is that this phagocyte contains pockets of enzymes, and those enzymes are called lysosomes. And those lysosomes get attached to this phagocytic vacuole and they actually release enzymes. And when the enzymes are released, this pathogen is digested and destroyed. Please do not say killed. So, number one, the pathogen, the phagocyte moves out. The phagocyte moves out. The phagocyte, as it moves out of the capillary, please understand phagocytes can only move out of capillaries. Capillaries are the only thing which are one cell thick. They cannot move out of arteries or veins. So the phagocyte moves out of capillaries because it's attracted by that chemical called histamine, which was released by the damaged cells. Then as the phagocyte moves out, it comes in contact with all the pathogens, maybe even dust particles, maybe bacteria, maybe viruses, maybe fungal spores. So the phagocyte is a non-specific response. It just engulfs, releases enzymes, digests, and destroys the invading pathogen. And this is how your body is protected against invading pathogens. Now, we're going to talk about the lymphocytes and how they produce antibodies. And what are antibodies? Antibodies are proteins. And these are structures which are going to lock on to the invading pathogens. So the first thing that you've got to understand is whenever a virus, like a measles virus, enters your body, it's a droplet infection. So it'll be present in the air, and maybe somebody in your class has it and is going passing it out in the class, and you will inhale it and you'd get the disease. So virus. Now, what it has on its surface is its face ID, or we can say its surface markings. Now, the surface markings, in this case, I made these triangles, and these are the surface markings. And just like everybody has a peculiar face, your face is very, very specific, and when you look at it, your phone will open because of your face ID. So, antigens on its surface, and I sort of compare it with the face ID. Now, different viruses will have different antigen. Maybe there's a virus with this sort of an antigen on it. Maybe there's a virus with this sort of antigen on it. Maybe there's a virus with another type of antigen on it. So different viruses have different antigens on their surface, and that's why we call them the different strains of the virus. Now, when a virus enters, one of these lymphocytes is going to be selected. Why? Because it has antibodies on its surface which are complementary. So this is the antibody on its surface, which is complementary to this. So this is going to fit in here, and this lymphocyte is going to be activated. So when this lymphocyte is activated, it forms millions and millions of copies of it, and that is called cloning. So the first point is selection. When one lymphocyte is selected, then cloning, and then differentiation. It's going to differentiate into two different categories. So differentiation, which is a very typical word we use very often in biology. So now when this one specific lymphocyte is selected, which has an antibody on its surface which is complementary to the antigen. Now, millions of copies of these lymphocytes are going to be made, this specific lymphocyte. Now, some of these are going to be called memory B cells. Memory B cells mean they will remain in your system, maybe lifelong, in some cases only for a short while. And the other ones are called plasma B cells. And the plasma B cells, what they do is they P for plasma, P for produce antibodies. And antibodies are the things these which are going to stick onto the antigen. So this is an antibody. This is an antibody. This is an antibody. Now, when the virus is got all antibodies sticking to it, then we've got our biggest white blood cell, which is the hero in all this, and that is the phagocyte. And the phagocyte is going to engulf and digest and destroy this invading pathogen. So B lymphocytes produce antibodies. Now, antibodies are specific. So this antibody which sticks to this virus is not going to stick to any other virus, because that other virus is going to have a separate antigen on its surface. So different antigens, different antibodies, and that is what lymphocytes do, is they produce antibodies. Now, how do I explain tissue rejection to you? Tissue rejection is a phenomena in which we give you an organ from another person. Now, we have not as yet developed any expertise or technological advancement that we can grow your organs. Like, for instance, somebody's kidneys fail, so can we grow an organ somehow, somewhere? No, we can't at the moment. So, what we do is we do a kidney transplant. So, there are two people, person X. Person X, both his kidneys have failed, so he needs a kidney. Person Y is going to donate the kidney. Why is it going to donate the kidney? Because we've done a tissue match. A tissue match means that nearly the person X has this antigen on his every cell of his body has this antigen, because he'll be having the same antigens on his every cell of his body, because every human being starts from zygote, and the other person Y also has these antigens, but it's maybe slightly different. So we've done a tissue match. Now we're going to give this kidney to this person. But then what is happening? This person's lymphocytes know it. This is not my own body cells. So they start making antibodies against it and will destroy the kidney. So you have to give them immunosuppressive drugs. That's not a very nice thing to do, because then they have a cold, they will die of the cold. So you have to then balance out when to give them the immunosuppressive drugs, when to stop them when they have an infection. So this is the important point of tissue rejection. And person X getting a kidney from person Y, and then the lymphocytes of person X making antibodies against the kidney and destroying them. That is called tissue rejection. Why? Because person X lymphocytes know that this kidney is not my own. There might be a slight difference, but it's still going to detect it that this kidney is not my own. So let's make antibodies against it, because maybe it's an invading monster. But it's not an invading monster, it's a poor kidney which has been transplanted from one person to the other, because we have not as yet developed any method by which we can grow organs. So this is all what I want you to know about tissue rejection. Now the last syllabus point K is describe the transfer of materials between capillaries and tissue fluid. Now what you've got to understand is that a capillary is one cell thick. So I'm just drawing this capillary. I'm not drawing the holes in the wall. So this is a capillary. Now this side is the arterial end. The arterial end carries blood at high pressure, while the venous end carries blood at low pressure. So as the capillary has the arterial end, and this is the capillary, please remember. So a capillary has an arterial end where the pressure is high, and I'm going to just draw an arbitrary line, just like that, to explain something to you. There's no such line, actually. Now at the arterial end, what you have inside, you've got the plasma inside. And you've got the body cells on the outside, which I'm drawing in. I've changed the color. So the body cells are outside in blue, and they're on all sides. They're not only just one side, they're all sides. Now, when this fluid is inside, it's called the plasma. But when this fluid comes out and bathes the body cells, the same fluid which was inside the blood vessels now is outside. Now, this fluid is not going to be called plasma, it is going to be called tissue fluid. So the same plasma which was inside the capillary, this red is the capillary, and this plasma as it comes out, it is no longer going to be called plasma, it's going to be called tissue fluid. And this is going to bathe the body cells. This is going to bathe the body cells. So the tissue fluid is the fluid which is the water and the glucose and the amino acid and the ions, all that which has diffused out in all directions. So there's tissue fluid there, there's tissue fluid there, there's tissue fluid all around. So the tissue fluid is now surrounding the body cells. But then the important thing which you've got to understand is that we've got plasma proteins here. Plasma proteins. Now the plasma proteins say are 1,000. Now these cannot leak. So still here we have the plasma proteins which are 1,000. But then this side, the venous side, has less water because the water is all moved out. So as the water moved out here and the water moved out here, now the water is going to return. Why is it going to return? Because there's a lesser water potential inside the capillary, because the plasma proteins are soluble, large molecules. But there's much less water now because the water had diffused out here. So less water now here. If there's less water here, what does it do to the water potential? So it lowers the water potential because the solute is the same. So the water is going to return on this side. Water is going to return on this side. So tissue fluid being formed here, and then the water and the dissolved substances moving back into the capillary on the venous side. So most of the water moves back, but then there is some which does not move back. Now the water which does not move back has to go by an alternative path, and that alternative path is that of the lymph vessels. So there are, just like if you remember in the digestive system, we had the lacteals. We have this lymphatic vessels all over the body. And what is going to happen is the tissue fluid which did not return is going to enter the lymph vessels. So the tissue fluid which does not enter, but as soon as it enters the lymphatic vessels, then this same fluid is called lymph. So what you need to understand is the same fluid inside the capillary was called the plasma. The same fluid with the dissolved substances moving into the outside the capillary and surrounding the body cells is called tissue fluid. Then the same tissue fluid, which maybe is unable to go back into the blood, will return through another second method, and that is the lymphatic system. So the lymph vessels are then going to form a lymphatic system, which is going to carry back all this tissue fluid and will return it to the main circulatory system, and somewhere in the body will drain back into the blood, will connect with the blood vessel, and will carry all this lymph fluid into the blood circulation, and it will return this tissue fluid which was unable to return by another pathway, and it will be carried back to the blood. Now you can see here the lymphatic vessels, how they are intertwined with the capillary bed. And this is where the tissue fluid is going to enter them, and then there's going to be carried away. So the lymphatics from all over the body are going to join, and they're going to join, and they're going to then ultimately reach here and enter this blood vessel here. And the name of this blood vessel is called the subclavian vein. If you remember, clavicle is a bone which is called the collarbone. So there is a subclavian vein here where the lymph will drain. But I mean, I don't expect you to remember the name of this, but I want you to know is that basically all the lymph, which was all the tissue fluid, which was unable to return to the blood, is going to be returned by another alternative pathway, and that lymph is again going to make the volume of blood the same, the normal volume of blood, which is about 5 liters. So the volume of blood will be maintained. It can't be, the tissue fluid can't remain around the cells, because if it remains around the cells, that is going to cause edema. Edema is when there is swelling. When there is swelling, like a person will complain that their rings get very tight, so their fingers swell up, or their feet swell up, or their ankles swell up, so that they don't, their shoes appear very tight by the evening. So edema is a swelling which is going to happen because the tissue fluid is unable to return back and through the lymph channels, and then return that fluid back to the blood circulation. So this lymphatic system is a parallel system, but then it doesn't have a pump. So the vessels have to be in between muscles so that they can be pressed, and the lymph can be moved onwards. So another point that we missed out here in the B lymphocyte story was that the plasma B cells produce antibodies, but then we have another set of cells also which are called the memory B cells, and these will stay in our system because they will remember this pathogen, and whenever a year later or 2 years later, if this pathogen enters again, these memory B cells will become plasma B cells, produce antibodies, and destroy the invading pathogen. And that's what we do in vaccines as well. We give you these dead viruses or weakened viruses so that you can develop the memory B cells, and the memory B cells will help protect you against the disease in the future. So please understand this part of the lymphocyte, which is the production of antibodies and the production of memory B cells, which remain there to protect your body from the same pathogen next time when it enters your body. That finishes your chapter on transport in humans, O-level biology. Best of luck, and I hope you've understood this chapter and you are able to do the exam questions, the MCQs, and the other questions on this chapter. Thank you.